All right, so we've established that the regulatory regions of genes can change quite a bit, yet still maintain the expression of genes. Now, that's not always the case because obviously there is change in expression, and sometimes those result in new features and new benefits. So even though there is still remarkable flexibility where we can maintain the expression regions and domains of the genes, there are also examples where we have changes that then result in a change in the gene. And realize that those two elements are very intimately connected, right? That we can change the expression level of the gene, but if we change the function of the gene, that might have an impact on those, either the driving the change in them or maintaining the ancestral form of the regulatory elements. So regulatory elements can have an impact on the evolution of the gene for which they control, and the gene which they control can impact the evolution of those regulatory elements. So here's an example across a wide range of flies where we're looking at changes in regulation that do lead to some differences in the downstream product of this gene. And so notice that although the same two genes are controlling the expression of this downstream gene, right? So we have two upstream genes having an input into the expression of this downstream gene. And this downstream gene has different function in these flies. But it's still the same two genes that are controlling them, just with different regulatory elements, and so those interactions result in a downstream difference in this gene. And so we may have some compensatory changes that maintain integrity of this signal, but we can also have different mutations that impact and have a role in changing the maybe function is not the right word because it's still the same gene, maybe even the exact same protein coding sequence. But even though it's the same gene, it could have a different phenotypic impact. And so that regulation can have a dramatic impact on the function of the gene. And so there's variability in this regulatory DNA and in the gene expression. And our initial studies are showing that there's lots and lots of variability, that there's lots of variations, sometimes even in a single individual. And so that's really another field of biology. It's kind of evo-devo, but a little bit not. It's more of kind of individual variability and flexibility in expression levels, and we can still have successful species. But certainly as we get to the level of populations, now we're going to start to talk about the raw material to differentiate and build between subcategories in that population. And then once we get to speciation, that's what we're really looking at for evo-devo, is what levels there lead to differences in species. Okay, and so does this variation correspond with phenotype, or is there some other sort of buffering mechanism, maybe environment, that allows there to be the same phenotype of a trait controlled by a gene because there's some compensation in the environment or compensation at other sites? And the answer to this is, in short, we don't know 100%. We're still looking at that. So all the differences that we see in individuals, some of it is truly just down to allelic variation. But oftentimes we don't even talk about, especially in introductory courses, or even think about the role of gene regulation. And so the reason that individual A looks different than individual B may certainly be that they have different alleles. And that's an abbreviation, or that's a shorthand for it, and might even be the answer for most of the differences. However, there is a significant, if minor, input for regulation. So if you have the same exact alleles, but they are controlled slightly differently, or maybe even drastically differently, then you're going to see a very different outcome in phenotype. So certainly regulation does have some, and it's still unknown in most cases how much, but it still does some and probably a significant amount of work causing differences within individuals. Okay, so just to give you some background on our last example here, there's a paper done in 2003 that was looking at single nucleotide polymorphisms. Now, we've come across these before. They're abbreviated, sometimes called SNPs. These are simply differences between individuals in a population at one specific nucleic acid site. So we'd say at this site, on this chromosome, right next to this gene, 20% of the population has a T, a thymine, and 80% of the population has an A, an adenine. And that would be an example of a single nucleotide polymorphism. And so if we know across the entire population it's 20/80, we can use that as a marker, because if it is for a particular group, right, if it is not at that 20-80 balance, then we know there's something going on there. And the shorthand is that there's something near that marker that plays a role and defines the group that you are looking at that doesn't have the specific ratio of 20 to 80 or whatever it might be. Now, originally, this could be done with microarrays, where we were looking at gene expression levels across a subset of genes, and microarrays still play a role. But today, much more commonly, and in fact much more powerfully, we use QTL studies and entire genome sequencing to accomplish the same thing. So in 2003, using these microarrays, they looked at nearly 1,500 SNPs that were spread across the entire human genome. And of those, a good portion, because that was the focus of this paper, were in these transcribed regions within part of a gene. But so 500 of them were outside of that or were near genes, okay? And 602 of them were heterozygous, so that means we've got some difference. So we start with this big, huge data set, we look at 602 and we say, oh look, this means individuals carry 2 copies that are different of this particular SNP. And in 54% of those, there showed a preferential allelic expression, meaning if you have 2 alleles, and you're heterozygous for them, one of them gets made more than the other one. And that can be some kind of standard genetics things, right? But we don't often think about mechanisms of dominance, right? So you learn in early genetics class that, this allele is dominant over this allele. But what is the mechanism for that dominance? Does that mean that we only transcribe the dominant allele and the recessive one never gets transcribed? Well, that could be one mechanism, but they could both be transcribed, right? And in fact, it looks like at 54% of the cases, one of them is transcribed more than the other. And so that might be a mechanism of dominance or a mechanism of the genetics that controls that specific system. But that leaves 46% where the mechanism of dominance is something very, very different, right? Where they're both being expressed at the same level, and maybe some of them are codominant and they don't have a dominant recessive trend, but others are. And so realize that regulatory evolution can play a role all the way down into even just what we kind of just originally categorized as very basic genetics. Is it dominant? Is it recessive? Okay, well, it's dominant. But is it dominant because that's the only copy that's made, so that's what shows the impact? Or is it dominant because they're both made at equal levels, but the other one competitively binds with all the target sites much better? Or there can be many, many mechanisms. And for many of these, we don't understand them really well. And in fact, in 30% of these, one of them is made at a level 4-fold greater than the other. Okay, so anyway, lots of variation, lots of things that need to be investigated here. So let's talk about the take-home message in all these cis-regulatory regions. So this is not only from the Low et al. Paper, which if you're interested you can look at, but we just hit the highlights. But that there's variation in allele expression, and this is incredibly common. Okay, so that's a regulatory function. Epistasis, which is unpredictable or sometimes kind of unusual interactions when we have 2 different alleles, that does play a critical role, and we're only beginning to scratch the surface of epistasis in determining why individual A is different from individual B. And in humans, we're good at visually judging differences in many phenotypic traits. In fact, a lot of culture, for good and bad, and ethnicities and things like this, is kind of founded upon this ability for us to say, hey, that person is different from my people, right? And so we're really, really good at that. But what we become to recognize, particularly as we've sequenced DNA, but even as we've taken a more detailed, closer look at other species, is that other species are nearly as variable as humans. Some are just as variable, but humans do tend to have a high level of variability no matter how you measure it. But other species do have variability and variation. It's just we don't always immediately recognize it or recognize it as easily as we do for our own species. So diversity levels are high. And the take-home message is that some of that diversity comes from control of expression rather than the alleles themselves. Okay? So there is a degree of freedom in cis-regulatory elements. They can change and mutate, but still have the same expression. These regulatory elements are modular. They can be mixed and matched. We can duplicate them. We can move them back and forth, and thus result in an incredibly wide range of regulatory control for those genes. And then there are other combinatorial factors that we may need to play into a role. So for instance, an allele might have an impact on another allele. It might control its expression levels. And so those sorts of things can make it very complicated and very tricky. And still a lot of work needs to be done, certainly for specific cases. But even for us to get a more general overall kind of rule of thumb about how these things work, we would need to do a lot more work. And so this is ongoing stuff, and this is an area that's ripe for continued research. And it might be critical for understanding specific instances of, let's say, hey, Alzheimer's disease, why does this gene do it? Why does it do it in this genetic background but not this one? But there's still a lot of unknowns there. And so Evo-Devo is a very, very young science. We're still working on it. We're still trying to understand in model systems how everything works, because those are the ones that are most approachable. Then we're trying to figure out whether what we understand from the model system can be generally applied, how much of it can be generally applied across all organisms. And then we continue to work and expand work on different models, and maybe even look at organisms that we cannot use as model organisms, but are interesting and important for other reasons, humans being, of course, one of the key ones. And so for many of these questions, they still remain unopened, and there's still a lot of work that needs to be done. And so we're working on that. We're beginning to understand that at a better level, but still tons and tons of work. Many new techniques are adding to our ability to do this. CRISPR is just one of them, where we can do some very nice functional things with CRISPR, but there are other techniques and modifications of older techniques that are also giving us a dramatic revision. And so Evo-Devo is still a relatively young field. It's only been around for, really 30 to 40 years, and many parts of it, many tools that we use in modern Evo-Devo have only been around for a decade or less. And there are new experimental procedures on the frontier that will allow us to do even more. So there's still a lot of work and a lot of understanding that needs to be done for this. And then finally, as we understand more, some of the tried and true theories are being revised. The obvious one is the kind of overall theory that most evolution was originally thought to be changes in the protein sequences themselves, where it turns out that as much or, more, right, a significant majority of the changes that make the big differences are regulatory changes. Okay? And so we're continuing to work on that. Our last discussion for next time will focus on plants, but up until now we've just done animals. And so we just want to review, I want to review just a couple of major points here as we wrap this up and look at these trends that we see across animals. We can look at what's going on today between species and extrapolate what happened in the past. And sometimes if we've got a complete phylogeny, even if we know nothing about the expression of genes in extinct organisms, which we don't, right? We can't look at expression of those extinct organisms. We can make some very strong and very clear extrapolations based on what's happening today, and then looking at that in the phylogenetic context. We can take our model organisms and say things about all species on Earth to some extent, but we need to take these model systems with a grain of salt. Although that might be a general rule that applies to all organisms, say the way that cells divide, we need to confirm that and make sure. We can't just say, oh look, Drosophila does it this way, therefore species A, B, and C do it that way too. We need to make sure we confirm that and use model systems in their most powerful mode as a null hypothesis that may be generally applicable but needs to be tested before we can make certain about how widely applied that trend is. And often we see the exact same mechanisms that are responsible for small changes and for large changes. So Evo-Devo has reinforced this point that microevolution and macroevolution are not really separate things. They're just the same evolutionary forces expanded over larger spans of time. And then finally, this last assumption, the same mechanisms, we can look at how these things impact changes within a species and changes between a species. The stickleback example we looked at was a really good example of that. So our last lecture will be next week, and it's looking at plants